RNAi, short for RNA interference, is a powerful tool to knock down, basically reduce the levels of specific proteins by targeting and destroying the messenger RNAs that have the instructions for making those proteins. Because we're only targeting the messenger RNAs, not the gene itself, this is only going to be a temporary effect. We don't need to worry about kind of messing with the genome. You still got that original recipe. RNA interference takes place naturally inside of our bodies in the form of microRNAs, and we can also introduce it in the laboratory as an experimental tool, as well as a therapeutic, in the form of siRNAs, small interfering RNAs, or shRNAs, short hairpin RNAs. To understand how these all work and why you might choose one or the other, let's step back and think about how RNAi works. RNAi at its heart involves a protein called Argonaute and a small piece of RNA, about 21 nucleotides long, that Argonaute uses as a guide. We call it a guide because it's going to guide Argonaute to specific messenger RNAs that have complementary sequences. It doesn't have to match the entire guide, but there's a 6 to 8 nucleotide stretch called the seed, and that seed is going to bind to a complementary site on a messenger RNA. If you have more complementarity than that, so say you have full-length complementarity, then Argonaute, assuming that it's Argonaute 2, the main Argonaute that our body uses, Argonaute 2 can actually go and slice or basically cut that messenger RNA, leading to its degradation. If you don't have a complete match, which is the case with microRNAs typically, then what happens is that Argonaute is going to bind to cofactors, other proteins that are going to help bring in deadenylation complexes to remove the tail from the messenger RNAs that was protecting them, as well as decapping complexes and exonucleases that are going to chew up the messenger RNAs. And so you get reduced expression of the protein. Those microRNAs are made from long precursors called primary microRNAs. These are transcribed from genes, so made as if they were going to go make a protein, but instead of making a protein, they make a functional RNA that then gets processed in the nucleus by a complex called microprocessor that's going to cut it, and it's going to make it so you have this long hairpin that gets taken into the cytoplasm, and then it gets bound by a protein called dicer, which cuts it into a duplex. That duplex gets loaded into Argonaute. Argonaute ditches one strand, the passenger or star strand, and now it has a strand that it can go and seek out and destroy messenger RNAs that match. We can provide into cells that double-stranded RNA duplex, like Argonaute would have gotten from the processing of the primary RNA, that duplex that can get loaded into Argonaute. Argonaute ditches one strand, and so when we give it just as this short duplex, we call that like an siRNA duplex. However, that's going to be restricted to giving it just as a duplex, the cell's not making more of it itself. If you want the cell to be able to make more of it, say longer term, well, now you can introduce a short hairpin RNA. The short hairpin RNA is going to be made in the nucleus just like your primary microRNAs would, and so it's going to get processed in that nucleus, and then it's going to be transferred into the cytoplasm, be processed by dicer, loaded into Argonaute, and all is great. And so that is kind of the two different strategies, the siRNA duplexes or the shRNA. With the siRNA duplexes, here you're just giving it to them, and so it's kind of like feeding a man a fish, versus with the shRNA, here you're teaching the man to fish because the cells can make it themselves. You're giving them the genetic instructions in the form of the DNA instructions for making those guides. Now, you can do this either temporarily or more long-term. So for a short-term knockout, you can just do that double-stranded RNA, the siRNA duplexes. For medium term, you can just transfect or basically introduce into cells the genetic instructions for making it in the form of a plasmid, so kind of an extra chromosomal circular piece of DNA that has the instructions for making that, but it doesn't really have instructions for doing anything else. It's just other than replicating this plasmid. And so then the cells can make that, but then with every cell division, that's going to get diluted out because it's not getting integrated into the cell's own genome. If you want the cell to be able to make this longer term, you want a longer term knockdown, then what you can do is you can actually transfect the cells with a version that's going to get stably integrated into the genome. There are different types of like viral vectors and things that you could do with this, homologous recombination or even CRISPR, in order to edit the genome in order to make this longer term. However, you're not messing with the original version of the gene for making that protein still. You're just introducing the instructions for making something that is going to reduce the levels of the gene product. And again, it's reducing the levels of the protein by targeting the messenger RNA copies of the recipe for making the protein rather than targeting that original recipe itself. Now, with the siRNA duplexes or with the shRNA, in both cases, you have to worry about potential off-target activity, so basically targeting the wrong genes because of the not being enough specificity between the sequence of the seed and the sequences in the DNA, or because of activity of that passenger or star strand. So if you think about a gene, you've got double-stranded DNA. One strand, the sense strand, is the strand that's actually going to be going on to make the messenger RNA. It has the actual instructions for making the protein. And the other strand is kind of just like the other piece of the sandwich. And it has the complementary genetic information so that you can make copies of it. That complementary strand, that antisense strand, is the strand that actually is going to be what your guide is going to look like. However, in the duplex, you're going to have one strand that actually looks like the messenger RNA and one strand that looks like the piece that you want that complements the messenger RNA. And if that strand that looks like the messenger RNA is used as the guide, well, now it's not going to be able to bind to the messenger RNA. And it might be matching somewhere else in your genome and causing off-target activity, in addition to not causing what you want, the knockdown of that specific messenger RNA. So scientists have to design these siRNAs or these shRNAs in specific ways using various rules and testing out various things in order to make it so that Argonaute preferentially loads one of the strands versus the other. It loads the strand that you want and not the strand that you don't want. And so this involves a lot of computational stuff, a lot of trial and error, and often what you do is you would buy it through like a library that's already kind of had these validated for knocking down specific genes. So you don't have to go through that whole process yourself. There's another cool way that can be used to reduce the levels of the passenger or star strand getting used, and this is to use a miR-451 mimic. miR-451 stands for microRNA 451, and it's this weird naturally occurring microRNA in our bodies that is involved in making blood cells. And why it's weird is that instead of getting processed by dicer, it actually gets processed, that second cut happens in Argonaute itself. So it still gets made in the nucleus, but the hairpin that results from that initial processing has a shorter top, and so it's not really compatible with dicer, and so instead it binds straight into Argonaute, and Argonaute does the cleavage of one strand. Because Argonaute is cleaving the one strand, it doesn't have to worry now about choosing which strand of the duplex to load because it's kind of already loaded the right strand. And so in this way, you reduce the passenger strand activity because that passenger strand is always going to be that same strand and Argonaute's not having to choose. So scientists found that you could actually adapt that seed sequence of the miR-451 because remember, it's that short sequence that actually matters, that actually is going to be used to do that initial binding. And so by changing that sequence, then scientists can transform this miR-451 into a platform for targeting different messenger RNAs. And so this is actually the strategy that was used in the recent announcement of a Huntington's disease treatment for targeting the defective gene for the Huntington protein in patients with Huntington's disease. And this was really exciting because the disease was typically untreatable in terms of actually treating the root cause of the disease and slowing progression of the disease. And so this platform can theoretically be used to target a variety of different things. It's a lot easier to use RNAi in the laboratory than it actually is to use therapeutically, however. This is because you actually need to get the RNA into cells, you need to get it into the cell types that you want, because it's going to have this kind of shorter term effect, especially if you're not integrating it into the genome. Now you need to worry about potentially administering dosages of it and getting those dosages safely into the places that they're needed. And so strategies for doing this include things like lipid nanoparticles in order to do that delivery. With siRNAs in particular, you really need to worry about making sure that these get to their location safely without the body's immune system kind of like being, oh, there's double-stranded RNA that might indicate there's a virus, let's go and attack it. Let's use exonucleases, chew it up, things like this. And so you want to use modifications such as backbone modifications that make the RNA more sturdy and things like this in order to reduce that sort of immune response and that non-specific response to having the short RNAs. The problem is you need to make sure that you don't make it too foreign that the body can't recognize and process it in the way as if it were the duplexes for Argonaute to use. Argonaute's really particular too in that it needs to be able to bind the ends, so you can't do a lot of modifications on the ends, which is where you would normally go to protect RNAs. Another problem with getting RNAs into places where you want them is that you want to make sure often that you're just getting them into specific cell types and that you're getting them into that cell type. And so you can do things like add modifications that are going to target, say, the liver by adding specific sugars that will bind to receptors on the surface of the liver. So there are a bunch of different strategies that can be used to try to target these RNAs to specific locations. Again, with the siRNAs, you're actually giving the RNA itself, but with the shRNAs, here you're typically giving the DNA instructions for making the short hairpin RNAs inside of the cells themselves. So here, instead of dealing with stabilizing the RNA, you're dealing with just delivering the genetic instructions. And so here you're thinking about things like different transfection techniques, things like maybe using a harmless virus in order to deliver those genetic instructions, rather than using something like a lipid nanoparticle in order to deliver the RNA. So lots of different strategies to think about, but at its heart, they all involve Argonaute and the small RNA. So that could be a microRNA, it could be an siRNA, it could be an siRNA coming from a short hairpin RNA or from a miR-451 precursor. Argonaute is going to use that small RNA as a guide to find and bind to messenger RNA targets that have complementarity to at least a 6 to 8 nucleotide long region of the guide called the seed sequence. When Argonaute binds, if you have full complementarity, Argonaute can slice that target and then the target gets chewed up. In other cases where you don't have full complementarity, Argonaute calls in cofactors. It binds to this protein GW182 or TNRC that's going to basically bring in deadenylation complexes to remove the tail, decapping complexes to remove the cap. Things are going to get chewed up. You can also get translational repression where the translation stops, but the mRNA doesn't really just get destroyed. All these things are going to lead to a knockdown or a reduction in the levels of the protein by reducing typically the levels of the messenger RNA for making that protein. So Argonaute is awesome. RNAi is awesome. It's just a little complicated when you think about how to make sure that you're doing it in the way that is going to target specific things, not target other things, and actually make it to where you want it.